Alright, so this video is going to um, go over the introduction of cells, the different structures of cells, different functions of um, the different structures and organelles within cells, and kind of the importance of studying cells in general. Um, Cells are pretty darn important. They are the structural and functional building blocks of all organisms, right? They're the smallest unit of living matter. And as I was telling my students, we're all made up of cells, right? Cells kind of create the organizational hierarchy of living organisms, in that you have um, atoms that come together to form molecules, and the molecules will come together to form larger molecules or macromolecules, and then they can combine to create cells. And then from cells, you can have interacting or similar groups of cells to come together that form tissues, and then from there you get organs, and on and on and on. So cells are the kind of, again, the structural and functional building blocks for living organisms, and for the different tissues and functional structures of organisms. Now, cells were first described back in the 1600s. I think it was 1665, by an English physicist, Robert Hooke, in his work called Micrographia. And this work ushered in the science of microscopy and confirmed its importance in biology. While he was examining a thin slice of cork plant, Hooke observed these rectangular units composing its structure, and he called these units cells, because of their resemblance to monk's chambers. So from there, we had the rise of cells and cells, kind of the concept of cells, and then its furthering importance in the biological sciences. When we talk about cells, we talk about cell theory. So cell theory, again, is this kind of concept in which the cell is the basic unit of life. And there are three main tenets for cell theory. First is that cells are kind of the smallest structures that perform the processes essential to life. So again, they're the basic unit of life in all living things. These processes that they perform can include food consumption, waste production and disposal, cell creation via reproduction, a number of functions, and each cell takes on specific tasks. You think of brain cells, muscle cells, skin cells, blood cells, they each have a specific task that plays a specific function in the body. So this is the process of cell differentiation. So they're the smallest structures, they're the basic unit of life. Each one performs certain processes and functions that are specific to organismal functioning. The second tenet is that all cells arise from the division of pre-existing cells. So we start out as one cell, right? We start out as a zygote formed from the fusion of sperm and egg, and that then divides into billions and billions of other cells with identical DNA. So all cells arise from the division of a pre-existing entity, a pre-existing cell. And then third is that, again, every organism is made up of one or more cells. So again, this goes into the concept of cellular organization, right? Many atoms create many molecules, and then molecules will come together to form macromolecules. Macromolecules come together to form cells, and then from cells you have the formation of tissues, organs, and then organ systems. So those are the three major tenets of cell theory and cell biology. Now, all cells, there are four kind of similarities or components that are synonymous across all types of cells. All cells have kind of these four basic components. First is a plasma or cell membrane, and that surrounds the cell and separates it from the environment, whether it's the external and internal environment. Second is a cytoplasm. This is a substance between the cell membrane and region where the DNA is held. It's made of cytosol, which is this jelly-like material that fills the cell. It includes cell components such as organelles, macromolecules, ions, and different filaments. All cells also have genetic material. Right? Usually this is DNA. It's some sort of genetic material that's made up of nucleic acids. Some cells utilize RNA as their genetic composition. Most and foremost use DNA. And then lastly, all cells have ribosomes, which are organelles that function in the synthesis or function to synthesize proteins. Now, okay, so yeah, all cells share these kind of same four basic components, but there are also several major differences. And these differences account for two broad categories of cells, or two broad categories in which cells are grouped into. And those are prokaryotes or eukaryotes. It differs kind of in regards to their cellular composition and where the chromosomes are actually located. So we'll start with prokaryotes. So prokaryotes are predominantly single-celled organisms or unicellular organisms. They're very simple. And these were kind of the first kind of forms of life during the evolution of actual life forms. They followed suit after protocells, which were the early cells to exist on Earth. And prokaryote cells, they refer mostly or occur mostly in bacteria and archaea domains, but they're known for being highly adaptable. They're able to survive in these very harsh environments, which is why they were so compatible living in the early Earth ecosystems and early Earth systems. Prokaryote, so the name pro means before, kary means nucleus. So prokaryotes, simple way to remember them, they don't have a nucleus, or they don't have a true nucleus. Instead, their genetic material is kind of coiled up in this central region known as the nucleoid. And that nucleoid is not surrounded in a membrane envelope, hence why it's not referred to as a true nucleus. Now, these guys are generally much smaller than eukaryotes, and again, a lot simpler. So, as I said, they don't have a nucleus or a true nucleus. Rather, their chromosomal DNA is concentrated in the nucleoid, which is this central region here. And then this purple material represents the actual bacterial chromosome. They have no membrane-bound organelles. And again, they lack a lot of, because they don't have those membrane organelles, they lack a lot of the structural complexity of eukaryotic cells. Going over some of the parts of your prokaryotic cells, again, most of these are bacteria and archaeal and domain. Most will have their cellular material enclosed in a capsule that's made of polysaccharides. So the capsule is this kind of yellow sheath surrounding this prokaryotic cell. And the capsule enables the cell to attach to surfaces in its environment, so it kind of acts like an anchor of sorts. They also have a peptidoglycan cell wall, which acts as an additional layer of protection. So that's this layer here. And the cell wall, it acts as protection, but it also helps to maintain shape and prevent dehydration, which is very important for these cells. Some of the material can pass through the capsule and it can cause desiccation of these cells. Once the cell goes into desiccation or dehydration, you lose cellular functioning unless it's able to produce an endospore, which some bacterial species are able to. But that's not kind of a universal trait. Some prokaryotes will also have these additional structures on the exterior, flagella. So these are these long kind of tail-like structures that are used for locomotion. Some will have pili. Pili are used to exchange genetic material during conjugation, which is improperly referred to as bacterial mating. They're not mating, they are transferring genetic material, but it doesn't involve sex organs. One that's not pictured here, but sometimes are included, are structures called fimbriae. They're used a lot by bacteria to attach to a host cell, and they can also use fimbriae to attach to different surfaces to enable colonization. So this is a structure that, again, allows for the rapid colonization of different bacterial species in a host or on a surface. So those are your prokaryotes as far as their structures. Again, they're pretty simple in comparison to eukaryotes. We'll get more into eukaryotes in a moment, but before we move on, one point that I had mentioned was that your prokaryotes, in addition to being simpler, they're also much smaller than eukaryotes. And when I say they're much smaller, I mean they are much smaller. So prokaryotes are usually range in size from 0.1 to 0.5 micrometers in diameter. You compare this to eukaryotes, which range from 10 to 100 micrometers. So why would it be beneficial, or why would what would be the importance of the small size in prokaryotes? Figaro, don't play with your toys. Well, with prokaryotes, again, they lack these complex structures. They lack organelles. And in order for them to, or their kind of mode of nutritional transport is diffusion. So by having a small size, it allows ions and organic molecules to quickly diffuse into the cell and waste products to diffuse out of the cells. With eukaryotes, it's different. They have developed structural adaptations to enhance intracellular transport. Prokaryotes lack those adaptations because they don't have organelles. So they're able to afford this small size because they lack organelles and intracellular complexity. And because of that, they need a smaller size in order to have more rapid diffusion across their cellular surface. Now, kind of again going into this, why be smaller rather than larger? Again, it falls on the same notion. Kind of going into this concept of the surface area to volume ratio. As surface or I should say, as a cell increases, its surface area to volume ratio decreases. Um, so for each kind of square micrometer of membrane, only so much of a particular substance can cross per second. So you can actually increase that surface area to volume ratio as you decrease cellular size. And that's the goal for prokaryotes. They want to be able to have as much surface area as possible for diffusion, but as small of volume. So they want to increase their surface area, decrease their cellular volume. They can decrease their cellular volume by decreasing size. So in general, an increase in surface area means you can absorb more from the environment. So you have an increase in diffusion, which allows faster transport of materials. Based on the kind of the notion of the surface area and volume ratio, as you decrease cellular size, you can increase that ratio, meaning you'd have a higher surface area relative to volume. So that enables prokaryotes to have faster diffusion without maintaining a larger volume. So it's faster and more rapid diffusion across a larger surface area relative to their actual volume and size. And again, that's important because this is the main mechanism for for intracellular transport in prokaryotes. Again, they lack the structures that enable them to, or kind of the structural adaptations that eukaryotes have that enable them to pump materials. Now, a sufficiently high ratio of surface area to volume. This is especially important in cells that exchange a lot of materials with their surroundings. So you can think going into eukaryotes, looking at certain cells like intestinal cells. These cells, along with prokaryotic cells, can actually create certain adaptations that enable them to increase their surface area without appreciable increases in volume. So for example, your intestinal cells have these long, thin projections that extend from their surface called microvilli. And these extensions allow for them to increase the surface of absorption, but again, being able to do so without increasing their volume. And prokaryotes will adapt or use kind of similar notions in their structures, and we'll kind of see this trend going across or continuing across as we go through different types of cells and think about where they're placed in the body and their function in the body. So this isn't just something that's kind of boxed in for just prokaryotes. You see this largely with prokaryotes, but you also see this notion of the surface area to volume ratio holding through for different types of eukaryotic cells based on their function. All right, so we talked about prokaryotes and kind of how they're different from eukaryotes. Let's start talking about eukaryotes. So we'll focus more on eukaryotic cells for the remainder of this lecture series, because we'll be discussing the different organelles and each of the functions of those organelles in eukaryotic cells. Now, eukaryotes are primarily and predominantly multicellular in nature. There are some that are unicellular, but again, the majority are multicellular. So your eukaryotes, as kind of a recap, these include animals, fungi, plants, and protists, right? Those are the four kingdoms in the domain Eukarya are Animalia, Plantae, Fungi, Protista. So All four of those are known as eukaryotes. They all are eukaryotic cells. Eukaryotic cells have a membrane-bound nucleus, so they have a true nucleus, and they also have membrane-bound organelles. The membrane-bound nucleus of each eukaryote contains chromosomes, which are pieces of DNA that are tightly folded up into proteins. And the remaining substance of each cell consists of the cytoplasm, which we had talked about before. This includes the cytosol and different organelles and particulates that are suspended in it. Now, as we go through the organelles, the best way, or the easiest way for me that I found when I was going through school to remember the organelles was, and their functions, was to kind of assign these community roles per se to the organelles. So each one, I'm able to remember the function of the organelle by assigning it some role that you would see in thinking of the cell as a municipal township per se. And by giving those kind of little descriptions or kind of little tasks per se to each organelle, you're better able to break down and remember what the overall function is. Again, this is a lecture topic that a lot of students get overwhelmed with just because there are so many organelles, and they try to remember every single little detail, which is important, but it's also kind of a quixotic, meaning it's irrational. It's not real or a realistic approach to the subject matter. So just kind of consider that, and you'll see that come into more play as I go through each organelle. But before we do, eukaryotic cells are broken up into your plant cells and animal cells. They have several similar structural similarities per se, but they also have several differences which serve to distinguish between plant cells and animal cells when you're looking at them through microscopes. So the main there's kind of two major structures that separate, actually three major structures that separate plant cells from animal cells. The first is this organelle that's called a central vacuole. And we'll get more in depth of what that's used for, but usually when you're looking at plant cells, you're able to determine this is a plant cell because it has this giant blob in the middle of each cellular unit, and that's not the nucleus. That's usually this central vacuole that's used to hold water, and it will grow and shrink in size depending on the amount of water that's allocated into it by the cell. Another structural difference between plants and animal cells: plant cells have cell walls. So animal cells do not have cell walls. This usually blows people's mind. They're like, what? Like, I thought all cells have cell walls. Animal cells don't. They have something instead called an extracellular matrix, which is more of this fibrous latticework structure that's covers or is held outside of the plasma membrane. And it allows, or it will connect into different areas of the plasma membrane and allows for a lot of intracellular communication and kind of flexibility in the cell, but it also functions with identification. There are some immune functions related with it. So it's more of this latticework fibrous network that doesn't necessarily provide a huge element of structural support per se. It's more of this intracellular communication. And we'll get more into that when we talk about cell walls and cell membranes. So plants have cell walls. Animal cells do not. They have the extracellular matrix instead. Plants also obviously have chloroplasts, right? So plants photosynthesize. Chloroplasts are the photosynthetic organelles of your plant cells. Animal cells do not photosynthesize. Unfortunately, we cannot just stand out in the sun and suddenly create energy, which would be awesome. We don't have that functionality. So we do not have your chloroplast structures. Some things that, so right down here I have what you don't find in plant cells but can find in animal cells. So plant cells won't have lysosomes, and they don't have centrioles, which are kind of structural filaments per se. And they have more of, they have some of their intermediate filaments, your microtubules and microfilaments, but they don't actually have true centrioles. And centrioles is what you appear during different stages of cellular division, just kind of keeping it simple. We'll go into mitosis and meiosis in other lectures, but right now I want you to kind of just focus in on just these cell structures for now. So animal cells, and they look a bit different. So your plant cells have kind of these rectangular cuboidal structure, or this hexagonal shape. When you look at plant cells, it looks like a honeycomb. And each cell actually, while we're here, in the there's cell wall and kind of that outer wall in between each cell. They have these intracellular junctions that are called plasmodonta, or plasmodesmata. They are this amazing intracellular kind of transport system that connects every adjacent plant cell to their neighbors. So it allows the entire plant, or kind of all these cells, to become a continuous unit. So the cytoplasmic material of one cell is shared with the cytoplasmic material of all the neighboring and adjacent plant cells, which is again really neat. And you can see that in this these kind of honeycomb like structures of the plant cell. When you put a dye into one cell, you'll see it actually disseminate into all of the neighboring cells because of these plasmodonta, and then it actually spreads throughout that entire kind of cross section of plant cells. This is why when you put salt down, the entire blade of grass will die. So in the winter, we put salt down on the side of the roads, or we put down salt on the roads and then it runs off to the side. And when you look at the side of the roads, all the grass and vegetation that lines the road is brown. It's dead. Sorry, I have a frog in my throat. And the reason for that is that salt breaks down cell walls. So when it's absorbed in the water that the plant material takes in, it travels into a cell, it breaks down the cell walls of that cell, but then because of these plasmodonta, it's able to travel out through the entire plant. So the entire plant will then die, because you have the kind of this dissemination of this salty fluid, or semi-fluid, semi-solid material throughout the entire plant. And then as it spreads throughout the plant, it breaks down all of the cell walls. So you have the rupturing of the cell, and the release of its cytoplasmic material and organelles. All right, so back to animal cells. Again, They have a much different appearance. They're not going to be these kind of cuboidal structures like your plant cells. They don't have the plasmodesmata structure, so they have different means of intracellular junctions. They use tight junctions and desmodenta and other types of kind of joint junctions per se. As I said, they don't have chloroplast, they don't have the central vacuole, and they don't have cell wall. Some things that your animal cells do have that aren't often seen in plants, they do have flagella. So flagella sometimes appears in plant cells. It depends on the type. Some plant cells will, their sperm cells will have flagella. They have centrioles. They have the same microfilaments and microtubules, but again, on top of that, they have the centrioles. And what was the major one? Oh, and as I said, they have the lysosomes, and we'll get to those in a moment. But the major difference is that animal cells do not have a cell wall. Again, they have that extracellular matrix instead, that kind of lies external to the plasma membrane. All right, so let's actually start going through kind of some of these organelles and understanding their differences or their functions. Organelles, the name means little organs. So organelles are essentially little organs that have specialized functions, and they allow different functions to be compartmentalized in different areas of the cell. So it's kind of this specialization feature per se. All right, so let's start with the organelles that are involved kind of in the control of genetics, or the cell's genetic control. These two organelles would be the nucleus and ribosomes. We'll start with the nucleus. The nucleus, again, as I said before, when I was going back many years through schooling, I assigned kind of these different communal roles to the organelles in cells to kind of help remember their function. So the nucleus you can think of as the mayor of the cell. The nucleus houses the genetic material of the DNA. Most of the genes are stored in the nucleus. There are some genes that are stored instead in the mitochondria and chloroplast, but again, the majority of the genetic material of the cell is contained in the nucleus. And then the nucleus ultimately functions in cellular regulation and production. It's enclosed in a double membrane structure that's called the nuclear envelope. Again, this is a double membrane, so it has an inner membrane and an outer membrane. The nuclear envelope separates the contents of the nucleus, so it separates the DNA from the cytoplasm of the cell. And each layer is kind of this lipid bilayer that then also includes its associated proteins. But the envelope itself is perforated with these little pores called nuclear pores. And these contain protein structure called pore complexes. So that those pore complexes, or those protein structures, line the surface of each pore and helps to regulate the entry and you can kind of see them right here. Helps to regulate the entry and exit of different particles. Now within the nucleus, the DNA is kind of organized into discrete units called chromosomes, which again are the familiar structures to us that carry genetic information. Each chromosome is made up of chromatin. So this is what you can see here, labeled as chromatin, your uncoiled DNA. Chromatin is a complex of proteins and DNA, and they're these thin strands, basically your uncoiled DNA that will coil up and then clump up before cell division. So before cell division, it looks like a giant mass of spaghetti, and then they will condense to form separate structures of chromosomes, which we're familiar with. And again, each eukaryotic species has a specific number of chromosomes. In human cells, for instance, we have 46 chromosomes in the nucleus. 23 of those chromosomes are for sex cells. On the other hand, fruit flies have 8 chromosomes in their nucleus, with 4 occurring in the sex cells. So again, each eukaryotic species has a specific number of chromosomes, again, that are held in the nucleus. Another pertinent structure of the nucleus is this little bulbous structure here, and that is called the nucleolus. The nucleolus is also known as this pupil of the eye structure. It appears as this mass of densely, darkly stained granules. And its role is to aggregate ribosomal RNA. So it actually serves or functions in the production of the large and small subunits of the ribosome. Once those ribosomal RNA units are kind of synthesized in the nucleolus, the proteins are exported from the imported into the cytoplasm and are assembled to form the actual ribosome, and they exit the nucleolus and the nucleus through those nuclear pores. So that's what the nucleolus is for. It's for the synthesis and aggregation of ribosomal RNA, which will be used to create the large and small subunits of the ribosome. We'll talk about the ribosome in just a moment. So as I said, nucleus, its major functions, it houses genetic material and regulates cellular structure, and it produces the ribosomal subunits. So the ribosomal subunits are produced in the nucleolus. Genetic material is housed in the nucleus itself. And the last component of the nucleus is the nucleoplasm. The nucleoplasm is this semi-solid fluid inside the nucleus, and that's where you actually find the chromatin and the nucleolus. So that's your nucleus in a nutshell. So that's the first organelle for involved with the cell's genetic control. The second one would be your ribosomes. So ribosomes are kind of the catalyst of protein synthesis. They are what I refer to or think of as protein factories. They are made up of ribosomal RNA, or your rRNA, and proteins. And again, that they are the specific organelles that will carry out protein synthesis. Protein synthesis will either occur in the cytoplasm or in the endoplasmic reticulum. Now, the number of ribosomes that are found in a cell will generally increase um, in cells that kind of have higher rates of protein synthesis. So, for example, you'd see a higher amount of ribosomes in kind of the cells of the pancreas that secrete digestive enzymes for the breakdown of different molecules and compounds. And as I said before, there are two locales where protein synthesis can occur. They can either occur in the cytoplasm or in the endoplasmic reticulum. So that causes kind of the consideration or the classification of two different types of ribosomes. Ribosomes can either be free ribosomes or bound ribosomes. Free ribosomes are those that are suspended in the cytosol. So these are the ribosomal structures. Your free ribosomes are the kind of catalytic mechanisms or factories that will create and synthesize proteins in the cytoplasm. And these generally are proteins that are used inside of the cell. So, for instance, a lot of your enzymes. The second type are bound ribosomes. Bound ribosomes are those that are attached to the outside of the endoplasmic reticulum or the nuclear envelope. Generally, they're attached to the rough endoplasmic reticulum. And these will synthesize proteins that are to be transported out of the cell. So for going back to the example of the cells in the pancreas, the cells of the pancreas have a high proportion of bound ribosomes because those enzymes are utilized in packaging, exportation, and secretion. So again, your bound ribosomes generally make proteins that are specialized in those same functions: packaging, exportation, and secretion. Now, the ribosomes can alternate between these two roles. They're kind of identical in structure. So you could have bound ribosomes switching and acting as free ribosomes in the cytoplasm, and then rebinding to serve as bound ribosomes. So that covers your nucleus and the ribosomes. Again, they're the structures for controlling the genetics of the cell. We're now going to move into this separate system, which is known as the endomembrane system. The endomembrane system itself is not like a single organelle. Rather, it's a system that's comprised of several different organelle components. The purpose of the system is to regulate protein traffic and perform metabolic functions in the cell. So it serves to modify, transport, package different lipids and proteins. And as I said, the system itself, it's not just one organelle. It's a kind of this network system of different organelle structures, and it's actually composed of 5 specific structures, or 5 specific organelles. These are the nuclear envelope, endoplasmic reticulum, Golgi apparatus, lysosomes in plants or vacuoles in, lysosomes in animals or vacuoles in plants, and the plasma membrane. The plasma membrane is included because it interacts with these endomembranous organelles. Now, the components of the endomembrane system, they kind of form this system and are related to one another either directly or indirectly. So they're either related through this direct physical continuity of their membrane layers, or it's more of this indirect connection via the transfer of membrane segments in tiny vesicles or as tiny vesicles. Vesicles are just kind of these sacs that are made of outer membranes. So again, the whole system again is composed of these 5 organelles, and it's because these organelles are related to one another, either directly because they have their membranes are continuous with one another, or they're indirectly related through vesicle transfer. So let's move into some of these organelles of the endomembrane system. So the first structure we'll talk about is the endoplasmic reticulum. I think of this as the highway, kind of as the functional, community functional role of this organelle. The endoplasmic reticulum is a huge network of membranes, and it actually accounts for more than half the total membrane present in the cell. It can again, these networks of membranes kind of exist as tubules and sacs that are called as cisternae. Under these kind of tubular structures here, cisternae the word refers to a reservoir for fluid, and these kind of look like little troughs in structure. Now, the endoplasmic reticulum, the membrane, separates the internal compartment of the endoplasmic reticulum, which is known as the lumen or the cisternal space, from the cytosol. So the lumen is the open space, or kind of the actual trough component within each cisternae. Now, because the endoplasmic reticulum is continuous with the nuclear envelope, the space between the two membranes of the envelope is continuous with the lumen of your endoplasmic reticulum. So that's how those two are connected. They have a direct continuity because of the connection of their membranes. Now, there are 2 different regions of the endoplasmic reticulum that differ in structure and function. These are the smooth endoplasmic reticulum and the rough endoplasmic reticulum. The smooth endoplasmic reticulum is kind of this tubular branch, like it almost looks like a coral reef structure on the outer edge of the endoplasmic reticulum. And it's named because its outer surface lacks ribosomes, so it has a very smooth appearance. Now, this component, it functions in the synthesis of lipids, also functions in the metabolism of carbohydrates, as well as detoxification of different drugs or poisons. So it contains enzymes that are crucial to those functions. For example, in the liver, hepatocytes, which are your liver cells, they contain and produce enzymes that are manufactured kind of in the smooth endoplasmic reticulum that help to detoxify different toxins. Some other functions of the smooth endoplasmic reticulum, it stores calcium ions. So this is important for muscle cells. In muscle cells, they have a kind of a specialized smooth endoplasmic reticular membrane that pumps calcium ions through the cytosol into the lumen of the endoplasmic reticulum. And when a muscle cell is stimulated by a nerve impulse, these calcium ions rush back across the membrane of the endoplasmic reticulum and move into the cytosol, and that's what helps to trigger the contraction of the muscle cell. So that's the smooth endoplasmic reticulum. The other type is the rough endoplasmic reticulum. So the rough endoplasmic reticulum is named because of its rough appearance, and that's caused by the fact that the membrane, or the outer surface of the membrane for the rough endoplasmic reticulum, is studded with ribosomes. So the rough endoplasmic reticulum has ribosomes. Smooth endoplasmic reticulum does not. Now the ribosomes that are kind of bound to the rough endoplasmic reticulum, these are your bound ribosomes. The bound ribosomes are the ones that stud that outer surface to give the rough appearance. Now the rough endoplasmic reticulum is its major function is modification, production, and shipping of proteins, and that's why I refer to it kind of as the highway. Many different specialized proteins are produced by the bound ribosomes that are attached to the rough endoplasmic reticulum. And when these bound ribosomes start to synthesize these proteins, it begins to create a polypeptide chain. That chain will grow from the bound ribosome and is kind of threaded into the lumen of the endoplasmic reticulum through a pore that's kind of formed by this protein complex in the membrane. And as that new protein, that new polypeptide, enters the lumen, it'll fold into its native conformation. So again, this is it going into the concept or the functions of the rough endoplasmic reticulum, the fact that it will facilitate polypeptide folding for proper modification and production of different types of proteins. And then these proteins, again, many of them are these secretory proteins, will depart from the endoplasmic reticulum in these little vesicles that will bud out kind of like bubbles from a region called the transitional endoplasmic reticulum, which is this area here. So you have the budding of the membrane to form a transport vesicle, and that will transport these protein products to other organelles, more specifically to the Golgi apparatus. It can also transport them back to different areas of the endoplasmic reticulum if they need to, if those proteins need to be remodified. Now, kind of a cool aspect of your rough endoplasmic reticulum, it's kind of this self-growing structure. You can think of it as not only a protein factory, but also, a protein factory or production house, but also it serves as a kind of a membrane factory, in that it will grow in place by adding membrane proteins and phospholipids to its own membrane, which is pretty interesting. So that's the endoplasmic reticulum. After leaving the endoplasmic reticulum, remember I talked about these little transport vesicles that are budding off the transitional ER. Once they leave this site, they travel to the Golgi apparatus. And the Golgi apparatus I think of as the post office in the community. It serves as the warehousing, sorting, and shipping center of the cell. So here, products of the endoplasmic reticulum are modified, stored, and then sent out to other destinations. The organelle itself is pretty neat in structure. It consists of flattened membranous sacs, which are, again, called your cisternae, to kind of give this appearance of a stack of pita bread. The actual numbers of stacks or number of cisternae in this structure ranges from kind of several hundreds to thousands. And these are kind of unlike the cisternae of the endoplasmic reticulum in that these are not physically connected. So each one is kind of this individual, independent unit. With the endoplasmic reticulum, it's more of like this maze complex where each cisternae is interconnected with one another. So you kind of have this continuous lumen created throughout. Again, Golgi, each one is a separate individual component. So they're not physically connected. You don't have the continuation or this continuous lumen track through the organelle. Now the Golgi stack, or the Golgi body, as we can call it, has distinct polarity, meaning that the membranes of the cisternae on opposite sides of the organelle differ in thickness and molecular composition. And it kind of creates two poles per se. The two poles of the Golgi stack are called the cis face and the trans face. So these act as the receiving and shipping departments. The cis face is kind of the receiving side, located near or closely situated to the endoplasmic reticulum. The trans face is on the opposing side. This serves as kind of the transport, or not transport, more of the shipping center. So little transport vesicles coming from the endoplasmic reticulum, they'll move that material from that organelle to the Golgi apparatus, and when they approach the Golgi apparatus, the vesicle can actually add its membrane and contents to the lumen of the cis face of the Golgi body by fusing with the membrane of that cisternae. So kind of coalesces to form a new cisternae in the Golgi apparatus. Again, going back to your trans face, this is the shipping size. This will give rise to new vesicles, which pinch off and travel to other sites. Now, this structure is not static. The Golgi cisternae actually follow what's known as the cisternal maturation model, in that the cisternae appear to mature moving from the cis side to the trans phase while carrying along these different proteins. And the products or the protein products in the cisternae are usually modified during that transitional movement from cis to trans. So the cisternae, again, they appear to mature. They actually progress forward, again, moving from cis to trans phase. Now, before the little vesicles are kind of dispatched from the trans phase of the Golgi apparatus, it will sort the products and targets kind of for various parts of the cell. And this targeting aspect is done by the placement of or addition of molecular identification tags. So the Golgi apparatus will actually tag this kind of the sorting component of it. We'll tag different products and we'll add molecular identification codes such as phosphate groups, for example. And that aids in sorting these products and determining or targeting specifically where they need to go in the cell. You can kind of think of these molecular identification tags as like zip codes on mailing labels. So the products are sent to the Golgi apparatus where they are sorted, tagged, and then shipped from the trans phase. So again, they're received at the cis, shipped out from the trans. So that's kind of the overall action of your Golgi apparatus. Again, what I refer to as the post office. All right, so we'll move into lysosomes. Remember, lysosomes are one of the structures that are found in animal cells only. You don't find these in plant cells. In animal cells, lysosomes kind of serve what I think of as the function of trash collectors. These are kind of the digestive aspects of the cell. It is a membranous sac of hydrolytic enzymes that animal cells will use to digest macromolecules that are taken into a cell and then recycle intracellular materials. Most of these lysosomes are made by the rough endoplasmic reticulum. So the protein products that they include, or the hydrolytic enzymes, are produced by the bound ribosomes in the rough endoplasmic reticulum, and then they're transferred to the Golgi apparatus for processing. Most of lysosomes will carry out intracellular digestion through a process called phagocytosis. This is a process where they engulf smaller organisms or other food particles. So here you can see a nice diagram. You have the formation of a food vacuole from that kind of buds off of the plasma membrane of the cell. So it's collecting some macromolecular component that is going to be utilized as nutrients for the cell, and it's packaged in a part of the plasma membrane that then buds off to form a food vacuole. Now, the lysosome will recognize the food vacuole and will fuse with the food vacuole, and those hydrolytic enzymes that are in the lysosome will then begin to digest the food products. The digested products will then pass into the cytosol and become or serve as nutrients for the cell. So it's essentially you're breaking down these macromolecules into their monomer components, and then those monomers can be utilized for beneficial and different specialized functions in the cell, or can be used to make different nutritional components that can be better utilized for the cell. Your lysosomes can also carry out autophagy. So this is kind of why they're called suicide sacs as well. This goes more into their recycling function. Lysosomes will use their hydrolytic enzymes to recycle the cell's own organic material. During autophagy, a damaged organelle or small amount of cytosol becomes surrounded by membrane, and a lysosome will fuse with that vesicle, and then the lysosomal enzymes digest and dismantle the enclosed material and kind of break it down into individual organic monomers. And those organic monomers can then be returned to the cytosol for reuse. So essentially, the lysosomes allow the cell to continually renew itself. Just as the lysosomes will recycle cellular material, if the lysosome itself becomes damaged, it will digest and break down its own structure, hence the name suicide sacs. So again, all of this allows for the continual renewal of the cell. All right, so animal cells have lysosomes. Plant cells, instead of lysosomes, they have vacuoles. And they actually have several different types of vacuoles. They're very similar to lysosomes, but they can carry out many more functions, or many different types of functions. There are food vacuoles, which are similar to those that are formed from phagocytosis, like I had previously described. Many freshwater protists have something called a contractile vacuole. So this is actually found in some of your freshwater eukaryotes. But the contractile vacuoles pump excess water out of the cell, thereby helping to maintain a correct balance of salts or minerals to fluid content. Now, going into this component, your central vacuole, I had discussed this a little before. This is a component that's unique to plant cells, and it's not found in any animal cells. This is a large pocket that's kind of nested within the cell, and it's usually the largest component of the plant cell. Again, as I said before, it's kind of the quick source of identification for identifying plant cells. Now, the central vacuole is made from kind of the amalgamation of, or merging of, several different vacuoles into one large unit. And it functions in the absorption of water. And by doing so, it contributes to plant growth by causing the cells to expand. And the structure itself is enclosed in a membrane that's called the tonoplast. In addition to water, the central vacuole will also reserve important organic and inorganic compounds. So such compounds like proteins, potassium, even chloride. And there's also other functions. So these are just some types of vacuoles. There are many other types. Many plants will use vacuoles kind of as disposal sites for metabolic byproducts, especially byproducts that could be harmful if they accumulated in the cytosol. So those are kind of taking on that kind of the lysosome property per se as these trash collectors. Some vacuoles in plants contain pigments that help to color the cells. So, for example, the red and blue pigments of flower petals that help attract pollinators. These are stored in vacuoles, more kind of pigment vacuoles in plants. Some vacuoles can also serve as defense mechanisms in that they contain toxins or impalatable compounds known as tannins, which help deter herbivores. So it serves as a anti-herbivore defense system. So these are vacuoles. Now, organisms, kind of one of the basic principles of living things is the ability to harness and use energy. Now, organisms will transfer energy that they acquire from their surroundings in an effort to utilize that energy. And this is done through two different structures. In eukaryotic cells, mitochondria and chloroplasts are the organelles that serve to convert energy into usable forms for the cell. All right, so we'll start with the mitochondria. Mitochondria I think of as the power plants of the cell. They are the sites of cellular respiration, which is the metabolic process that generates ATP by extracting energy from sugars, fats, and other fuels with the help of oxygen. The number of mitochondria that are present in eukaryotic cells is directly dependent on kind of the cell's level of metabolic activity. So you see higher amounts of mitochondria in such cells as muscle cells or cells of the liver, where you have a higher amount of metabolic activity occurring. The mitochondria are enclosed by two membranes. Each membrane is a phospholipid bilayer that has a kind of its unique collection of embedded proteins. Now, the outer membrane is this smooth structure. The inner membrane is more of this convoluted structure with these inward foldings that are called cristae. Now, the importance behind this, it actually goes back to the concept of the surface area to volume ratio. So having, or by creating these inward folds, it actually increases the surface area through which cellular respiration can occur without increasing the volume of the mitochondrial unit. So the cristae act to give that inner mitochondrial membrane a larger surface area for proteins and thus increase the productivity of cellular respiration. And the reason why this is important is because ATP synthesis takes place on the inner membrane. This is where ATP synthesis occurs and actual cellular respiration. So by increasing the surface area, you can increase the productivity of cellular respiration. Now that inner membrane also divides the mitochondria into kind of two internal compartments. The first is the intermembrane space, which is actually the narrow region that kind of separates or is held between the outer and the inner membrane. The second compartment is the mitochondrial matrix, which is actually enclosed by the internal membrane. The matrix contains enzymes, mitochondrial DNA, and ribosomes, and also has the enzymes that are responsible for ATP production. So mitochondria are present in both plant and animal cells. Another kind of productivity or respiratory organelle per se is the chloroplast. Now, your chloroplasts are only present in plant or algae cells, and they are the sites of photosynthesis. They convert solar energy into chemical energy by absorbing sunlight and using it to drive the synthesis of organic compounds, such as sugars from carbon dioxide and water. It's a specialized member of plant organelles called plastids. You see different types of plastids in plants. So there's amyloplasts, which are these colorless plastids that store starch or amylose in roots and tubules. There's also chloroplasts. Those are the ones that are most important here, in that they contain the green pigment chlorophyll, along with other molecules that are crucial to the photosynthetic production of different sugars. Now, the contents of the chloroplast are partitioned by an envelope that consists of at least two different membranes, and the two membranes are then separated by a narrow intermembrane space. Inside is a membranous system that kind of occurs in the form of these flattened, interconnected sacs called thylakoids. They kind of look like little pancakes, per se. In some regions, they're stacked together to give these structures or create structures called granums. The granums are, again, these stacks of thylakoids, and they actually look like a stack of IHOP pancakes. The fluid outside of the thylakoids, that's called the stroma. That contains chloroplast DNA, ribosomes, and enzymes. So those are kind of the three major compartments or spaces that are created by the membranes of the chloroplast. All right, so continuing on along here, we'll go into another organelle called peroxisomes. These are oxidative organelles. They're roughly spherical in shape and often have this granular or crystalline core that's considered to be this dense collection of enzymes. They have a specialized, or they serve as a specialized metabolic compartment that's bounded by a single membrane. So they don't have a double membrane covering like many of your other organelles. It's just a single membranous layer. And it will actually, the peroxisomes will cooperate with your mitochondria and chloroplast and plant cells to carry out certain metabolic functions. The major importance of the peroxisomes is that they contain enzymes that transfer hydrogen from various substrates to oxygen, which can produce hydrogen peroxide as a product, but they have the enzymes uh, necessary to break down hydrogen peroxide so that it does not become toxic to the cell. And some peroxisomes will use oxygen to break down fatty acids, and those fatty acids can then, once they're broken down to smaller molecules, can be transported to the mitochondria and used as fuel for cellular respiration. So they play a major role in kind of oxidizing certain substances, again, dissociating or breaking down hydrogen peroxide, byproducts, and fueling cellular respiration. All right, so the cytoskeleton. The cytoskeleton plays, or the major role of the cytoskeleton is kind of a structural support of the cell. It's composed of three major types of fibers. There's microtubules, microfilaments, and intermediate filaments. Intermediate filaments are more of these permanent fixtures of the cytoskeleton, and they're specialized for bearing tension on the cell. And each type is actually constructed from a different subunits of keratin proteins. Your microfilaments, these are solid rods that are approximately 7 nanometers in diameter, and they're built from a globular protein called actin, which is why they're often referred to as actin filaments. Their structural role in the cytoplasm is to bear tension, kind of the pulling forces, similar to the role of the intermediate filaments. They're also well known for their role in cell motility and contraction, particularly as part of the contractile apparatus of muscle cells. These filaments also function in amoeboid movement. So a cell, your amoeba cells actually are able to crawl along a surface by extending and contracting into different extensions, which is a function called pseudopedia. And that arises from the contractile and motile functions of microfilaments. The third type are your microtubules. These are found in the cytoplasm of all eukaryotic cells. So as I had said before, all of these cytoskeleton fibers or filaments are in both plant and animal cells. They are in all eukaryotic cells. More specifically, they are found in the cytoplasm of all eukaryotic cells. Your microtubules are basically comprised of this wall of a hollow tube, or that they're more of this hollow tube, and the wall itself is constructed from a type of globular protein called tubulin. Each tubulin molecule exists as a dimer structure, which means it consists of two slightly similar but just slightly different polypeptide subunits. The tubulin dimer itself is composed of the alpha tubulin and beta tubulin polypeptide subunits. And it will grow in length by adding these tubulin dimers to the ends of its kind of hollow tube structure. The function of microtubules really is in shape and support of the cell. Also serves kind of as tracks along which motor proteins can move, and is responsible for the separation of chromosomes during cell division. So this takes a closer look at each of those filament types of the cytoskeleton, kind of looking at their main function as well as their structure and protein composition or the subunits that compose each one. Now, one of the structures I had mentioned were centrioles. These were only found in animal cells, not in plants. Microtubules themselves grow from centrosomes, which is a region or in a region that is located near the nucleus. That's kind of considered to be the microtubule organization center. The centrosomes function kind of as the compression resisting grinders of the cytoskeleton. Within each centrosome is a pair of centrioles that lie perpendicular to each other. And each one is composed of 9 sets of triplet microtubules that are arranged in a ring. So here are your triplet microtubules. You have this arrangement of 9 of them that are kind of arranged in this circular shape, and this gives you the kind of the cross section of one of your centrioles. So this is an individual centriole, and then you'll have these centrioles kind of organized in an array to produce, or you actually have two centrioles that are kind of held perpendicular, so at a 9-degree angle, and those are then held in this spherical structure or cylindrical structure that kind of looks like a chimichurri per se, or chimichanga, whatever those cinnamon sticks are called. But so you have the arrangement of these centrioles to create the kind of the centrosomes. The centrioles themselves help organize microtubule assembly. And again, only the centrosomes of animal cells have centrioles. Plant cells lack centrioles. Now, some of these kind of specialized arrangements of your microtubules to form centrioles, some of this is responsible for the motion of flagella and cilia, which are locomotor appendages that protrude from some cells. They can also be used for propulsion, or to move kind of fluid over the surface of the cell. So they're used to give rise to these locomotive propulsive structures. They kind of take on this kind of sweeping motion. All right, so the last component, what are we missing? What structure have we not covered? Well, we have not covered the cell wall or the plasma membrane, which are both very important. So we'll start with the cell wall. I'm actually going to move backwards here. So kind of moving away from the interior of the cell that we've just toured, talking more about these external structures of the cells that also provide additional important functions. So in plants, you have cell wall structures. With animal cells, you have extracellular matrices. Now these structures, the structure of the cell wall and the extracellular matrix, these are extracellular components that kind of have their own structure, but also have intracellular junctions and connections between cells that serve to coordinate cellular activities and assist cell communication. Now, the actual methods or the types of these intercellular junctions are going to differ between plant and animal cells because they differ in the structures of those extracellular components. Again, in plants, the component are your cellular wall structures, and animals, they're extracellular matrices. So let's tour kind of the cell wall of plants first. The cell wall is, again, this structure that serves as a major distinguishing factor between plant and animal cells. Again, plant cells have them, animal cells do not. In plant cells, the cell wall serves as a protective mechanism. It helps to protect the plant cell. It maintains its shape. It also prevents the excessive uptake of water. Kind of looking at the level of the whole plant or organism, the strong walls of your plant cells enable the plant to be held upright against the force of gravity. Now, the cell wall is different from the plasma membrane. They are two separate entities, so don't confuse the cell wall with the plasma membrane. Again, those are two separate entities that differ greatly in composition and design. Now, with the cell wall itself, the composition may differ between species and cell type, but the basic design is the same. Most of your cell walls are composed of cellulose. So when we had talked about macromolecules, I had talked about cellulose being present in cell walls, and that's the structural polysaccharide that helps function in making these cell walls so strong and durable. They also have other types of polysaccharides, as well as different types of proteins, and they will kind of create this ground substance or matrix that is very, very strong. The cell wall itself is relatively thin and flexible, as well as being strong. It will first lay down or be secreted when plant cells are very young. And once the plant cell matures and stops growing, it'll begin to strengthen its cell wall. So you have kind of the deposition of polysaccharides and protein layers into that ground matrix to strengthen and solidify the wall. As I talked about before, with your kind of the plant intracellular junctions that can connect different plant cells together, are the plasmodesmata, or plasmodonta, as I like to accidentally and mistakenly call them. But your plant cell walls are perforated with these channels that are called plasmodesmata. As I was saying before, that's what allows or kind of unifies the plant as one living continuum. The cytoplasm of one plant cell is continuous with the cytoplasm of its neighbor. This enables water and small solutes to pass freely from cell to cell. So that's the cell wall of your plants. With animal cells, again, they don't have a cell wall. Instead, they have the extracellular matrix. So although animal cells lack quote-unquote cell walls, they do have this elaborate structure of the matrix. The kind of main ingredients of the extracellular matrix are glycoproteins. There are 3 different types of glycoproteins that are present. You have collagen, proteoglycans, and fibronectin. The most abundant glycoprotein is collagen, which forms strong fibers outside of the cell. And often these are embedded in a network of kind of interwoven proteoglycans. The complexes that are formed again contain hundreds of these different glycoprotein molecules. So you can have really large proteoglycan complexes that are kind of covalently attached to one another, and then also attached to long polysaccharide molecules. And then the fibronectins, which was that other type of glycoprotein, is what kind of connects or attaches cells to the extracellular matrix. They bind to cell surface receptor proteins called integrins, which are built into the plasma membrane. And they kind of will serve in transmitting or integrating changes between the extracellular matrix and the cytoskeleton. And that's why I was talking about the role of that, or the fibers of the extracellular matrix help to function kind of as this communication center, in that they are all linked together, and that linkage can transmit stimuli between the cell's external environment and its interior, and that can result in changes in cell behavior. So it's more of this fluid latticework structure that enables different changes to be made in behavior based on the stimuli that are received by the fibers and then transported by those same fibers of the extracellular matrix. Now, in animal cells, they also have intracellular junctions. The junctions are different from those of plant junctions. So plant junctions were the plasmodesmata. Animal intracellular junctions are tight junctions, desmosomes, and gap junctions. And we'll talk about these more when we talk about cellular transport. All right, so the only component that has not been covered are the plasma membranes. However, we'll stop at cell walls for this lecture and use kind of plasma membranes as the topic for the next lecture, which you can continue watching. It'll move right into that lecture, or you can watch at another time. But I hope this lecture helped you to understand or kind of better place the different functions of the organelles in different cells and the kind of the overall differences in cell structure. And again, remember to use those little kind of easy memory kind of tactics that I talked about, assigning kind of these community roles to the different organelles to help you or kind of kickstart your memory as to what their function is. It really does help. So I hope that you were able to take away a lot of information from this lecture. If you have any questions or comments, put them in the comments section below. And I hope that you walk away learning something that you didn't know before.